So let me begin with one simple question to all of you before we go for protein sequencing. How many of you have heard about DNA sequencing? Please say yes in the chat if you have heard about DNA sequencing. So many people, right? But did you hear about protein sequencing so often? Probably you would say that not so often. You might have heard, maybe due to your own requirements, like some of the university programs or maybe your competitive exams. What may be the possible reason that protein sequencing remained obscured? It was not very popular, although the idea of protein sequencing came uh, earlier, slightly earlier than DNA sequencing, because the time when DNA structure was deciphered, it was 1953 when Frederick Sanger and Watson and Crick, they came together with the DNA structure. But 2 years earlier, there was a famous paper by Frederick Sanger that elaborated protein sequencing of the insulin. And even before that, in 1947 and much before that, several people tried to sequence the protein. So, can you just tell me what was actually the need of protein sequencing at that time? Can anybody go in that period and then realize that what was actually the need of protein sequencing and why in due course of time it gradually became obscured? This doesn't remain a very popular method to do the sequencing.
So because in the earlier period, people thought that actually the reverse was happening. That is, protein is the main, genetic material.
Mhm.
And that's why they thought that protein sequencing was more important than DNA sequencing, I guess.
I would say protein sequencing was more important than DNA sequencing was never a thought. In fact, people did not knew much about DNA. But what you said is right, that protein was considered to be a major component of biological systems, because at that time, if you look at the history of biochemistry, you would probably know that all biochemistry started with the day-to-day processes like wine manufacture, sugar manufacture, and all the cheese manufacture. These were the major things, and a lot of enzymes were involved in that time. So the chemical nature of biomolecules was questioned a lot, and people thought that proteins are some of the most interesting and intriguing molecules which must be explored. Due to this reason, a lot of research was in early days done on proteins. In fact, if you look at the history of James Watson and Crick, so Watson also started his work in the early days by looking at proteins. So there was everywhere. The protein was in the air at that time, and everybody was talking about that. John Kendry was working on some of the protein structures of myoglobin and hemoglobin. Then the Linus Pauling was working on the helical models of protein. Even G.N. Ramachandran was working on collagen proteins. So protein was actually in the air at that time. But one important question that was in front of researchers, in front of scientists, that was what proteins are made up of and what is their exact sequence, what is the constituents of that. So already by some chemical reactions, chemists already depicted that, just give me a moment. Okay, so I was telling you the importance of proteins, that why proteins were important, and as you said, that proteins were some of the most critical molecules, biomolecules, and their identity, their chemical composition was needed in the early days, and that attracted most of the scientists towards deciphering that what is inside the proteins. So some of the early evidences, they showed that proteins are made up of amino acids, and as you know, amino acids are some of the molecules which contain an amino group and carboxy group together. And most commonly, we have alpha L amino acids. So, next important question that was in front of scientists was, are these amino acids present in specific order, or they are just present in random order? Is order of the amino acids is important? So almost during that period, there were two famous scientists. One was called Anfinson, and another was Leventhal, who were working on the protein biology, and they came up with the idea that protein structure is mostly governed by protein sequence. That was Anfinson's hypothesis, and there was a famous experiment which Anfinson performed, that the structure of protein is basically governed by its sequence. So in the 1940s, I would say since from 1920s to 1940s, there was lot of curiosity, lot of inquisitiveness among the scientists to know the sequence of a protein or sequence of amino acid within a protein, and that led to some of the remarkable discoveries of all times in biochemistry. And that is what we are going to discuss today, that how to sequence a protein. But eventually, what happened when the DNA structure was revealed and then DNA sequencing methods took over, then the genetic code was deciphered by Nirenberg, Mathai, Hargobind Khurana. All these people, all these people started. So those who are commenting about screen share, I'm not writing anything on the screen yet. It is the home screen which I projected in the beginning. So I will subsequently move on after answering some of the questions, creating a theme why we are looking at this thing. So don't worry about that. Listen to me what I'm saying. So what happened after that? Gradually, the structure of DNA was deciphered in 1953. Then Nirenberg, Mathai, Hargobind Khurana, all these people came up with a genetic code. And then the link between biochemistry, proteins, nucleic acids, all these things was well connected. And then what we know today as central dogma of molecular biology, that DNA has got some sequence of bases which encode RNA, which makes codons, and then those codons are kind of address for attaching specific amino acids. Then comes proteins. So gradually after that era, when proteins were known to be formed from DNA, the direction of research gradually moved towards DNA, and a lot of people started looking at the parent molecule. If you know about what parent molecule has, you can easily predict what its daughters or its children will have. So then started the era of somewhere in around 1980s. There started the revolution in DNA sequencing. So I would say somewhere around 1981 to 87, there were some landmark papers, and there was one connecting link, a person I would describe about, that is Frederick Sanger, who was a pioneer of both protein sequencing as well as DNA sequencing. And he is some of the rare people who got Nobel Prize twice. So after the discovery of DNA sequencing methodologies, people then quickly switched on to DNA sequencing. And as we know, if we are aware about the DNA sequence, we know about the codons, we can easily decipher the protein sequence, or we can predict the protein sequence. So that obscured this whole technology, and that did not allow this technology to become very, very popular. But there are instances where it is not exactly possible to predict DNA sequence, protein sequence from DNA sequence, because there is a difference in the codon usage across organisms. So it is not necessary that if you have a codon, you can always predict an amino acid. So in such cases, when there is a codon bias, when there is a codon usage ambiguities, in those situations, in order to precisely determine the protein sequences, you always have to go for sequencing of proteins. So still today, many people use protein sequencing as a independent technique, and they prefer doing that. So that's the objective of today's discussion, today's masterclass, and we will look at how to do protein sequencing in more details. So I'm now going to talk about what is the basic idea or basic theme of protein sequencing. So, unlike DNA sequencing is quite straightforward, because if you know about DNA, there are few simple things. That means DNA is made up of only four type of bases. There will be either A, there will be T, there will be G or C, and there is a fixed pattern. So, some of the very, very conventional and standard methods that you know, use of DDNTPs and termination of DNA replication that became very, very popular, which was pioneered by Frederick Sanger. So that chain termination method is quite easy, and now we know that a lot of automation has been done in that. So automatic machines are available which can quickly find out the entire sequence of the DNA by this method. But what is the challenge with protein? Protein sequencing may not be very, very easy. The challenge, it starts with basic number of units. Here you have just four bases to be recognized. So maybe you can tag them with four different things, four different fluorophores, and then you can use DDNTPs and easily find out the sequence. But unlike DNA, proteins have got much more complex environment. The first challenge there is there are 20 different type of molecules, 20 different amino acids, or if you want to add to that list, I can say 22 if you add selenocysteine and pyrrolysine as well to that. So that's first challenge. Then second challenge that comes in the way of protein sequencing is its higher order conformations. So when I say higher order conformations, that means there are tertiary structures, there are quaternary structures, and I would say there is also prevalent presence of disulfide linkages. So these two major challenges, I mean, first challenge is very large number of units, different type of units. So if you begin to design an assay or design a technique which can individually determine an amino acid just like it was done for DNA, it will be difficult to find out 20 different fluorophores which do not have any overlap in the emission spectra. So it's very, very difficult to develop a technique synonymous to DNA sequencing for protein sequencing. And then second thing that I mentioned, that there is a higher order conformation which is not found in DNA. So they can be of variety of shapes, and the problem becomes more difficult when there are multiple disulfide bridges. You have to break them and you have to identify them. So that particular challenge was one of the most difficult thing to develop a amino acid sequence in a protein. But this challenge was took over by two prominent people. I would show you their, I mean, pictures. I hope you can recognize this person, and I hope you can also recognize this person. So, can you name the first person? Anyone? We are talking about protein sequencing, so obviously he will be the pioneer of protein sequencing.
Protein Sanger. Sorry, Frederick Sanger.
He is Frederick Sanger.
Yes, yes, yes.
Yes, sir. And
First one. He's Edman.
Edman.
So these are the two pioneers who laid the foundation of protein sequencing, and the second person is also credited with discovery and development of methods for DNA sequencing as well. So everything that we are going to discuss today would actually revolve around their fundamental discoveries, what they made towards protein sequencing. So let's start looking at the overall process, how protein sequencing has to be done, and what are the different steps, because it is not as straightforward as DNA sequencing. In DNA sequencing, you just prepare your DNA, put it in the machine, or if you want to go for in the Sanger style, then you radio label it at one end, and then you use DDNTPs, then run a gel, and then start reading gel from the bottom, and you get the sequence. So that was the way of doing DNA sequencing. But in case of protein sequencing, we have multiple steps which have to be performed independently, and they have to be looked over separately. So let's look at the overall profile of how do we go for protein sequencing individually. So I would say the protein sequencing would actually start with purification of protein. You must have a pure protein sample, because if there are impurities, then you might get interference from other things. So the first step, step number one in the protein sequencing, will be purification of proteins, which can be done by a variety of methods. You may be knowing about them, like size exclusion chromatography or ion exchange chromatography, or maybe gel electrophoresis is also one of the protein purification approach. Now, as I mentioned, that proteins have higher order conformations, they might have disulfide bridges. So next important event that is required, or next important step that is required before the sequencing can proceed, is to denature them and to reduce them. So can you tell me how to denature a protein? You may be aware about that very well. What kind of denaturing agent you may prefer to
We
denature a
can
protein?
use urine.
Yeah, you can use SDS, you can use urea. There are many other examples like guanidinium thiocyanate. These are also other things which can be used for denaturing. Then what about reduction? Can SDS reduce or break disulfide bonds? Yes or no?
No.
No, sir.
No. So what should be done to reduce these things?
Beta
Beta
mercaptan.
Mercaptan.
Beta
Perfect.
marker.
Beta mercaptanol. There is another thing that is called as dithiotriol. Okay. Uh, but one problem here, when you do reduction, because generally what is known in from the textbooks and other things, we know that disulfide bond is created in the reducing environment. Can you please mute yourself, Vipin Singh? Your mic is creating disturbance. So disulfide bond is generally created in the reducing state. This is what we know. I'm sorry, it is created in the oxidizing state. And when you have a reducing environment, when you add any reducing agent, like as I mentioned, mercaptanol, and dithiotriol and other things, so this eventually leads to the breakdown of disulfide bridges, because there will be a lot of H plus or reducing agents available. So they will create this oxidized cysteines, reduced cysteines. Now, one thing that we do not know and we do not realize, that this process is readily reversible, because reducing agent cannot be present at all the times everywhere. There will also be oxidizing environment, since we do reactions in presence of oxygen in open environment. So if there is oxygen available readily surrounding the solution, then these whole things can quickly revert back. So if you believe that you have reduced your sample and now you are ready to go for sequencing, you are mistaken, because by the time you will begin the sequencing, it is likely that disulfide bonds can be reformed. So it is important to also prevent the reduction, prevent the re-annealing or reformation of the disulfide bond by the oxidation process. And that can be done by simple things like, I can suggest you something known as iodoacetate. So what iodoacetate does, these reduced cysteines, it connects with them and it acetylates them. So once they get acetylated, it is difficult for these disulfide bridges to revert back to the disulfide linkage form. So in order to completely denature a protein and then completely reduce a protein, it is important that you reduce disulfide bridges, and then after that you perform their modification by iodoacetate. So is this step clear? This was step number 2. Now, after doing this preparation, when you have done this, it is okay to go for sequencing. So now we have step number 3, where we go for sequencing. At the same time, when we do the sequencing, it is also important to characterize and to check the amino acid composition, because these sequencing methods may be error prone. They are just chemical reactions and there may be some chances of errors, or there may be some confusing results or ambiguous results where you might find two type of amino acids at the same position. So it is important to separately confirm that what is the amino acid composition of a protein, so that you have got an idea in the background. You know that protein has got these many lysines, these many leucines, these many tryptophans. So you can tally with your protein sequence and you can confirm that the sequence that you have found is correct or not. So a parallel step we have to do is amino acid analysis, and that generally begins with hydrolysis. Can you tell me what is hydrolysis? What is the difference between hydrolysis and denaturation? Anyone? Hydrolysis is breaking down the peptide linkages. Okay, But denaturation doesn't break down peptide linkages. So when you do hydrolysis, you split or you break down individual amino acids. You take off individual amino acids from the peptide, and you separately quantify, you separately find out that which are amino acids present in your solution. So the characterization of individual amino acids, or what we call as amino acid analysis, or AAA, that can be done with the help of a number of chromatographic methods. We will discuss little later. So that's another method. Now coming back to the sequencing strategy, so there are different type of methods. I mean, protein has to be written, and it can be depicted like a series of amino acids, and there will always be an amino terminal in a protein, and there will always be a carboxy terminal in the protein. So the idea of sequencing is that the basic idea that developed in early days, you will sequence one by one. You will take off this amino acid, you will modify it by some reagent, and then you will detect it. Then you will take the second amino acid, you will remove it, you will modify it, and then you will detect it. So this is how people go, and this can be done in either direction. You can either move from N terminal to C terminal, or you can move from C terminal to N terminal. So there are, you can say, two ways of sequencing. One can be N terminal to C terminal sequencing, or another one can be called as C terminal to N terminal sequencing. In short, they are known as N terminal sequencing methods, and the other one is known as C terminal sequencing methods. So you would realize that N-terminal sequencing methods are more popular, and they are more often used compared to C-terminal sequencing methods. Can anybody answer that question, that why in the initial days people focused more on N-terminal sequencing, why C-terminal sequencing was not very important and it's not considered important still? Anyone?
As translation
Covalency of carbon.
No,
from N to C.
your audio is not clear. Can you answer in the chat? N-terminal is unique. No, that's not the right answer. N-terminal is unique.
Sir,
What does
as
it?
sir,
Yes,
translation occur from N terminal to C terminal. That's why.
that's the right answer. So the correct answer to this question, that why it is important and why people look at the N to C terminal things, because protein synthesis occurs from N terminal to C terminal, and that's the fact you should know. Protein synthesis occurs from N to C terminal. Now, you may ask me that what if it happens from N to C terminal? I'm interested in knowing the whole sequence. So, yes, if you are interested in knowing the whole sequence, then it doesn't matter whether you do it from C terminal side or N terminal side. But generally, the starting part of the protein, it plays a very important role, N terminal part, in deciding that where protein goes, what kind of function it performs, how it folds. So a variety of features are available in N-terminal part of the protein. So in the early days when people started developing sequencing methods, they were mostly dependent on N-terminal sequencing methods. So are we now clear about this fact, that what are the different steps to be performed? So if you just have a recap of this thing, what we do is, step number one is purification of protein. Then step number two that we do is we denature the protein and reduce the protein. And in the step number three, we do the sequencing from either N terminal or C terminal. And parallelly, along with this, we will also be doing amino acid composition analysis. But that's not enough. Once you have broken down the disulfide bridges, it is also important to determine the sequence or position of disulfide bridges, because in order to completely know the sequence of a peptide, another major step that would also be present will be locating disulfide bonds, that where they are present. That's one more step. Now besides that, the whole thing that I described will be okay if you have small peptides. I mean, if you have just 10 amino acid peptide, then these steps are fine. But what if your peptide is, let us say, 100 amino acid long? So it is not possible to go on with these terminal methods or end methods. I will tell you in detail that why it is not possible to continuously do terminal sequencing for larger amino acids. Their sensitivity is gradually lost. As we go above 30 or 40 amino acids, it is not possible to determine the terminal sequences or sequence of the protein by these methods because the sensitivity is greatly lost. So what we have to do for the larger peptides, if there are larger peptides, then before step number 3, you also have to perform another step. I would mention that step as 2A. So what is that step? That step is known as proteolytic digestion. That is done by several known enzymes which can cleave at a specific position. So that is an additional step which we need to do because we want to fragment the protein first, and then we want to sequence the individual fragment, as the sequencing methods are limited by size. So we cannot sequence the larger sequence. So if there are larger peptides, we must have to break them down into smaller pieces. Now this adds to the challenge. If you have a larger peptide, if you break them up after the proteolytic digestion, because everything will be happening in a solution. So in a solution, there will be all different kind of fragments, and you will get variety of results of the sequence. So the biggest challenge in the end, I mean, after this step, which is locating the disulfide bridges, the last important step, or step number 6, would be to integrate the complete information. So whatever you have got from step number 4, that is acid hydrolysis, whatever information you got from proteolytic cleavage, whatever information you have got from Sanger sequencing or all these kind of terminal sequencing, position of disulfide linkages. So at the end of the sequencing process, in order to get a complete sequence of protein, what you need to do in the end is that you have to integrate that information. So that requires a huge amount of logic and aptitude to compile all these things. So are we clear about these basic steps, that how protein sequencing could be done? You purify a protein, denature a protein. If it is larger, then digest it with proteolytic enzymes, then go for any of the terminal analysis method. They can be either C-terminal methods or N-terminal methods. And then you parallelly you go for amino acid hydrolysis and amino acid composition. And finally, you determine the position of disulfide linkages, and then you integrate all this information. So that's an overview picture of how protein sequencing can be done. Let's move ahead now. Let's talk about specifically the step number 4. I already talked about how to purify protein and how to denature them. Let's now move on to the next step, that is step number 4, that is amino acid analysis. So what do you mean by amino acid analysis? I said it is determining composition. So if there is a peptide, like this is a peptide, it is denatured, and this is also reduced. So what you are going to do to find out amino acid sequence, as you could have seen in the previous diagram, that the step that I mentioned was hydrolysis. So fine, you go for hydrolysis. You can make use of caustic soda, that is NaOH, and you can heat it at a very extremely high temperature. And there are also differences. If your protein has got a lot of tryptophan and phenylalanine, larger amino acids, then it is also prone to hydrolysis. So you have to do more rigorous hydrolysis, or you have to do that for a longer period of time. So this is how you break down or hydrolyze a protein. Now when you hydrolyze a protein, you get individual amino acids that's broken down into individual amino acids. Now, can anyone tell me, how should I identify these amino acids? How would I come to know that is there any tryptophan or not? Is there any serine or not? Or if there are any serine, how many serines are there? How would I come to know that? Any idea?
We can tag a specific amino acid with a specific info, then we can detect.
Okay. What you can do? Please answer in the chat. I would prefer that much better because your voice is not clear sometime. So I suggest you, rather than speaking on the mic, you answer in the chat. My question was, how will you now come to know that which type of amino acids are present in a test tube where you have hydrolyzed a protein? Okay, so that's right. I mean, some of you are commenting that what has to be done. So it is basically chromatography that can help you out. And I believe almost every one of you would have done chromatography at some level in your higher education. And that was a very basic experiment that most of you did in your college time, where you were allowed to just add some dots or spots of mixture of amino acids. Then there was a thin layer of silica, or there was a paper on which you allowed these things to move. And gradually, your liquid that was present in the beaker, which was called as mobile phase, that allowed the movement of these amino acids onto this paper. And finally, in order to visualize those amino acids, what you did, you sprayed ninhydrin to it.
Ninhydrin solution.
Okay, because ninhydrin can react with amino acids and that can produce color. So after spraying ninhydrin to that sheet of paper, you were able to realize the location of individual spots. And then what you did after that, what you did, how you came to know that what is the identity of these amino acids? You probably determined a factor that is known as RF value, right? That RF value was defined as distance traveled by your analyte or amino acid, or the position of spot, and distance traveled by mobile phase. So you got a fractional value of RF, which was calculated. Suppose your mobile phase went up to this distance, let's say it was 12 centimeter, and the distance of one of the amino acid, let us say, was 6. So probably your RF value was 0.5. So what it indicates? How do you get RF value? If you know the principle of chromatography, this particular type of thin layer chromatography, you would notice that in this particular method, there is a partition of amino acids between the stationary phase in which they were dissolved and the mobile phase which is moving. So the amino acid which is more soluble in mobile phase, that will disperse first in that mobile phase and it will move fast, and the one that is less soluble will move slow or will remain behind. So based on that fact, if you consider a constant mobile phase for a specific mobile phase, every amino acid will also have a fixed or pre-known RF value. So that can be just formulated from a table, and you can match the RF values with the table. So if I find that 0.5 is the RF value of specific amino acid, say alanine, then I can figure out that this spot is representing alanine, this is representing serine, or this is representing tryptophan. So that's possible to figure out that which amino acids are present in your mixture.
ಅಲ್ಲಿ ಉಂಟು ಎರಕ್ಕೆ ಕಳ್ಸತ್ತಾನೆ.
Now, if I ask you another thing, that how would you come to know that there are 2 serine and 1 tryptophan and 5 leucine in the protein? How would you count the number of these things? Any suggestions? We can of course find out the identity, because every amino acid will have specific RF value. So any spot that is corresponding to RF value of any amino acid, we can figure out that which amino acid is present in the protein. Then coming to the quantities, relative quantities of the amino acids, how do we come to know about that? So I would say the density of spot would be an indicative of the quantity of amino acid. So if you have a spot, let us say leucine spot, which is 5 times as larger as the smallest spot on your TLC, then you can say this is present 1 times and this is almost 5 times compared to this. So density of spots can help you out to figure out that how many amino acids are present. So this is how crudely you can get an idea about the number of residues present in a peptide and the kind of amino acids which are present in a peptide. All right. So someone is asking, what if my protein is extremely hydrophobic residues? Is it possible then they would? Yes, of course. They will move, in fact, faster, because when you are doing conventional chromatography, the solution that you are using are mostly hydrophobic. So when you are using hydrophobic things, hydrophobic mobile phase, then hydrophobic proteins or hydrophobic amino acids will actually move faster. The problem arises when you have hydrophilic things. So when you have hydrophilic things, then you have to reverse the phase of chromatography. Instead of using hydrophobic mobile phase, you will have to use hydrophilic mobile phase, and your stationary phase need to be hydrophobic. So that's called reverse phase chromatography. Now this was just for the basic idea. I mean, this was just for explanation, but this technique cannot be very, very accurate, and there will be a lot of difficulties in analyzing them. So in the early days, people use these methods, but gradually, that chromatographic techniques also developed, and then we have some advanced methods like we know high performance liquid chromatographies came up. So instead of using the conventional type of chromatographic methods, the all this hydrolyzed fraction, this whole fraction, can then be fed into this kind of automated machine. So you can have a column for separation, then you can have a lot of amino acids, you can have a pump of ninhydrin that can give color to amino acids, and with the help of a buffer pump or a whole pressurized system, then you can allow these amino acids to individually move across a tube, and then as they will move one by one, they can be detected with the help of a spectrophotometer that is fitted in the machine. So rather than just manually looking at the spots, we can actually obtain the peaks of color or peaks of blue color, which can then be analyzed, that can be used for looking at the amino acid composition. So if you look at this picture, this might give you a brief idea about basic composition or basic trend that will be obtained when you run amino acids on a liquid chromatography. So that would be either high performance thin layer chromatography or high performance liquid chromatography. So these are the kind of techniques which can now automate the amino acid composition. And as you can see here, the retention time or the time at which the peak appeared can be different for different type of amino acids. You might see that arginine is showing the first peak somewhere around 10 minutes or before 10 minutes, like 7 minutes. Then you have other amino acids. You have several different amino acids like histidine here, and then you have tyrosine here. So what I mean to say, there can be every amino acid will have a unique position at which it will be eluted, because every amino acid has different properties. So this standard graph can be used to correlate with the sample. I mean, if you have a graph of all 20 amino acids, and if you know where they show a peak, at what time do they show a peak in a specific mobile phase, then you can put your analyte in the same mobile phase and you can check for the peaks. So if you, let us say, got only one peak, which is corresponding to methionine, that means it is only methionine that is present in your amino acid. Or if you get two peaks, one for methionine, another for leucine, then you can say there are only two amino acids. And the height of these peaks can help you to predict that what is the quantity or number of amino acids present in specific uh, protein. So that's how we analyze amino acids. That's how we do analysis of amino acids while we are doing protein sequencing. And that's the first part I wanted to discuss. So are we clear up to this level? We learned that the first step is you denature protein, you reduce protein, and parallelly you will do two things. One is the sequencing from terminals, and the second one is amino acid analysis. So right now we talked about analysis of amino acids. Now let's go to the next part, and that is step number, let me just take you to this part. I'm going to step number 3. I'll get back to step number 2a later. So I'm now going to step number 3, where I'm going to tell you that how to do terminal sequencing. I mean, when you have your peptide denatured and reduced, then how to determine the amino acid sequence from its ends, either from C-terminal end or from N-terminal end. So as I told you, there can be C-terminal sequencing or there can be N-terminal sequencing. And I mentioned to you their advantages, that N-terminal sequencing was very, very popular, and that became useful in the early days. So there are 3 important methods of N-terminal sequencing. The first one is known as Edman method. Second one, this was developed somewhere in 1947. Although Sanger started much before in 1941, but he was able to make it popular somewhere around 1951. Then there is another method called as densyl chloride method. So as I briefed you, the basic idea or basic theme of the protein sequencing from the terminals lies in a fact that you modify one amino acid at a terminal. You take an amino acid, you modify it, let's say you added some chemical group to it, and then what you did, you split it or you break off. You break only one bond, and once you have broken down one bond, your this particular amino acid is separated, which is modified, and then you detect it. Okay, so that's the fundamental. Now, what was the beauty and what was the work of these 3 famous scientists, 2 famous scientists, Sanger and Edman, that they explored some of the chemicals which can selectively react with N-terminal proteins. And they also developed and optimized the method that after modification of N-terminal, selectively you can cleave the N-terminal amino acid. Only one amino acid can be cleaved, and that can be analyzed. You already know how we can analyze it. If we have an amino acid already separated, then we can again go for the chromatographic methods, and we can analyze that easily, whether the first amino acid was tryptophan or tyrosine or phenylalanine. So that's the idea. Now let's start with the first principle that was developed by Edman. So there is a reagent which is known as Edman reagent.
अरे keep silent. Meeting चल.
Okay, so let me just tell you about what the Edman reagent is. Edman reagent is chemically phenyl isothiocyanate. So you can see here PTH, or some people also call it as PITH. So phenyl isothiocyanate here. Isothiocyanate means when it is connected through N. So phenyl isothiocyanate is a very important component which was discovered and developed by Edman. Now see what happens, what it can do. PITH has ability to react with a peptide amino terminal. So you can see an amino terminal here, and you can see there are lone pairs in amino terminal. They can attack this PITH carbon, and this whole group get conjugated. So this PITH complexes with protein and makes PITH protein complex. Now what happens after this when it makes a PITH protein complex? Remember that it happens at the pH 8. So at higher pH or alkaline pH, PITH can interact with a peptide, and that can make a PITH complex. I would highlight this PITH here. This is your PITH, and now this PITH has coupled with your peptide. It has got conjugated with your peptide. Now what we can do, an interesting step, we simply heat it in presence of acid. So if you heat it in presence of acid, this bond becomes very, very susceptible. The bond between the first next amino acid, this place. So I may highlight for you the amino acid or residue, this NH2R and this whole thing. This is one amino acid, okay? Whereas this is your next amino acid. So you can see here, this is your next amino acid, and this one is your first N-terminal amino acid. So where the cleavage is likely to happen on heating, this is the point. Here, this next peptide bond will be cleaved, and you will find that your PITC with the first amino acid is eventually cleaved, right? So you have peptide being released separately, and this first amino acid along with your PITH is separated, and this peptide is now cleaved off. So I think with the help of these color representations, it is possible for you to comprehend much better that what is happening when this PITH is reacting with it. So whole work of Edman was focused on this fact that there can be a reagent which can selectively interact with N-terminal, and after modifying the N-terminal, it is possible to break off the first amino acid. That was the only most important part of this method. So once you have separated that, once you have removed this first amino acid, that means PITH complex, so this becomes now, instead of isothiocyanate, it turns into thiocyanate. So this is thiocyanate amino acid complex, which is obtained in the solution. So what can be done? It can be then analyzed by chromatography. Okay, you can take your tube, suppose this was your tube and the reaction was taking place in the tube. You added everything, you did some addition of PITH at alkaline condition, then you heated this, and after heating you added some acid to it. Then finally what you did, you go for chromatography, right? So once you do chromatography, you will be able to determine, because your this part remaining peptide would be very large, and you can selectively detect this particular reagent by chemical reactions, functional groups. So in the chromatography, by having some PTH reactive groups, you can detect its position, and again by RF value, it is possible to exactly figure out that which amino acid was present at N-terminal. So I hope that should be clear to you. This is how the first amino acid on the N-terminal could be known. Now, you can repeat the same process. Your remaining peptide is intact. So in this tube, you can carry out the second cycle. You analyze it by chromatography, then you carry out the second cycle. You again add PTH, you again change the pH to 8, then you again heat. Then what you will be doing, you will be chopping off second amino acid. So you can go cycle by cycle, and after every cycle, you will be eliminating N-terminal amino acid, and you will be detecting it, right? So you add PITH, add pH 8, heat it, change the pH to acidic, you release free amino acid, and then you have remaining peptide in the tube. You can keep on doing this, and every time after every step, you can go for chromatography of that fraction, and you can detect that which amino acid is present. But now what happens, in this tube, there will be millions of molecules of peptide, right? There will not be a single peptide as I have shown in the figure. So it is not likely that all the molecules will be splitted in the first step, and in the second step you will get a perfect breakdown of the second amino acid. So you can have a number of peptides which will not be digested in the first step. Then you will again have number of peptides which will not be digested in the second step. So as you gradually cycle this from one step to second step to third to fourth step, the noise start to increase, because after every step, you will repeatedly get amino acids of the first position, amino acid of the second position, amino acid of the third position. So the drawback of this method was that you cannot go beyond 50 amino acids as the noise level increases to very, very high, right? Another drawback of this method was that detection system was not very, very sensitive, as detection of PITH is not sensitive enough. So in the parallel lines, another scientist's work, but let me just, before going to another scientist's work, let me show you the exact way how he did it. So actually Edman did not stop at this point. He came up with a very brilliant commercialization of this whole technique, and he developed a machine in which automatic protein sequencing can be done. So this was the original prototype of the machine that appeared in his paper. As you can see, this was the prototype of the machine which he developed for automated sequencing. He had all the chemical reactions to be done in that machine, and detection and everything could be done. And later on, it became very popular, and this was also commercialized, and this became famous as protein sequenator. Okay, so Edman is credited with development of protein sequenator, which is a protein sequencing machine. But as I told you, this remained obscured due to the fact that DNA sequencing rapidly emerged at later point of time. So if you would like to see how Edman did it and the chromatography plots, so this is how actually they looked like. As I mentioned, that after every cycle, he will be doing chromatography and detecting amino acids. So you can see here, this is, let us say, cycle number 1. So after first he got these pieces, then after second he got these pieces, third, fourth, fifth, sixth. So he did repeatedly all the fragments and then analyzed by chromatography. So that was really a meticulous job by which Edman was able to determine the sequence. And this paper, which appeared somewhere in 1966, that was a work on some of the proteins of sperm whale, which was related to hemoglobin. So this is how Edman worked. Now, in the same time, as I mentioned, in the parallel times compared to the Edman, another famous scientist, as I mentioned, who pioneered DNA sequencing as well, was Frederick Sanger. And what he did, he came up with similar idea, but that was more sensitive. And his idea was to change the reagent. Actually, he was in close contact with laboratories working in MRC, people working in MRC, Medical Research Council of UK, which is very famous for a lot of discoveries. So he came up with one new reagent, which was more sensitive and more accurate as compared to the reagent which was used by Edman. So the reagent that Sanger used is known as FDNB, and that is fluorodinitrobenzidine. So instead of Edman reagent, which is PITC, he used FDNB, that is fluorodinitrobenzene. And how it actually looks like, it's a little different than the reagent which was used by Edman. Edman's reagent was having NSH group, but this was having a fluorine here, this was having a NO2 here, and this was having another NO2 here. So this is known as fluorodinitrobenzidine, but the idea or the principle or logic was same. This reagent was capable of interacting with the first N-terminal amino acid. So as you can see here, this is your first N-terminal amino acid, and then you have next amino acid here, this is second amino acid. So instead of using alkaline and acidic system, which caused a lot of noise and problems in the solution, you can simply react FDNB with the N terminal, and then in presence of some highly acidic environment, you can make use of some highly acidic environments, like you can use hydrogen iodides. So in the presence of hydrogen iodide, it was possible to release HF from here. This fluorine could be released, and then this whole molecule, that is FDNB, was conjugated to your amino acid N terminal. So did you get the idea? I mean, principle is same, but here the reagent is different. Now, what is the advantage of this reagent? The ease of reaction, the sensitivity, and the accuracy of the results that you get. These were the advantages of FDNB over PITC that was used by Edman. So finally, he also did acid hydrolysis. In the presence of acid, it was possible to cleave this whole thing. So once you do acid hydrolysis, this FDNB was broken down along with the first amino acid, right? So this is how he got this. And then finally, what you can do is you can again go for the same process. You can go for chromatography. And after doing chromatography, it is possible to determine the first N-terminal residue. But unlike the first method, unlike the PITC method, which was developed by Edman, this method cannot be cycled again and again. So cycling is not possible here because during this process, the hydrolysis was so rampant that other amino acids were also broken down. Now you may ask me that if other amino acids are also broken down, will they not be interfering with chromatography? Will they not be detected in chromatography? So no, because this was having a specific color because of the nitro groups, and only this amino acid which is conjugated to FDNB, that was only identified, and rest of the amino acids were not identified in this process. So this was challenging to find remaining amino acids, because if you want to determine remaining amino acids, you will have to purify the protein which doesn't have the first amino acid from N terminal. Then you have to again go for the same process. But the conventional cycling, which was possible in the initial method of Edman, that was not possible here. But due to sensitivity and due to accuracy of this method, Frederick Sanger's ingenious work resulted in the complete deciphering of a sequence of insulin, which happens to be one of the very important protein in today's time. We know that a lot of clinical aspects of insulin are known. So he got the complete sequence of insulin by making use of this particular technique. And if you want to look at his paper, then it appeared somewhere in 1950s, I think. So these are the results of his paper. As you can say, there are plenty of chromatography pictures. So he took several fractions, and fraction by fraction, he collected all these things. And then at the end, he was able to predict that what is the exact sequence of insulin. So that was Sanger's work, and this whole work appeared in 1952 in the form of amino acid sequence of insulin, which became very, very popular discovery at that time. And that also led to a Nobel Prize for Frederick Sanger. So that was a Nobel Prize winning discovery of this person. And that's why he is credited with pioneering of this work. Later in 1980s, he also worked on DNA sequencing, which we are not talking today. So are we clear about this basic idea that how to do N-terminal sequencing, what is Edman's method, and what is the method developed by Frederick Sanger? Now, there were certain modifications that were done because the methods that were used in early days by Edman and methods which were used by Frederick Sanger, they were colorimetric, color-based. But as you know, color-based things are less sensitive. So there was another technique that was developed based on a modified reagent that is known as densyl chloride, or there is another variant which is called as debsyl chloride. So a third method that was later modified for the N-terminal sequencing is known as dipsyl chloride method, where another reagent, as you can see here, dipsyl chloride, it's formed by fluorodinitrobenzene. Two molecules are joined together. And as you can see here, this is quite bigger molecule with a characteristic SO2Cl group over here. So merit of this molecule is that it is fluorescent. And since it is fluorescent, you can perform the same process. I mean, you can tag the N terminal with dipsyl chloride, then you can do the acid hydrolysis, break down the N terminal amino acid, and then detect with the help of chromatography. But advantage would be, as it is fluorescent, so even less quantities of protein or less quantities of amino acids, they can be detected compared to the previous two methods that were developed by Frederick Sanger or Edman. So these were three prominent methods that were used in the initial days for N-terminal sequencing or sequencing of proteins from N-terminal site. Now, people also tried to look at the sequencing from C-terminal site, but they won't become very popular in due course of time, as I mentioned, because C-terminal sequencing is not very required. People generally look at what is present at the N-terminal site. So in order to do C-terminal analysis, some of the enzymes were used which can cleave it from the end. Like there are some proteases which are called as exoproteases or exopeptidases, which will cleave a peptide from its ends. So if you can find a peptide, find an enzyme which can cleave a peptide selectively at this point, and you can then analyze this amino acid, it will be possible for you to determine what is present, which amino acid is present at the C terminal. So one of such enzyme was discovered in later days, and that was known as carboxypeptidase. So by using carboxypeptidase, what people did, they incubated protein with carboxypeptidase for some time, and they knew that how much time is needed for and they knew that how much time is needed they knew that how much time is needed for only removing one amino acid. So they did that analysis after doing breakdown, but it is not as straightforward as you see. It's not like that only one amino acid will cleave and then you will detect this and then second can be detected, because carboxypeptidase will work in a way that it will break first. If it is cleaved, then they will cleave next, then they will also cleave next. So you will have a mixture of first amino acid, second amino acid, third amino acid, depending on for how much time you incubate a protein with that enzyme. If you incubate a protein with carboxypeptidase for a longer time, then probably your whole protein will be hydrolyzed. So timing is very important. When you are doing C-terminal analysis, you have to make sure that you have only chopped off one particular amino acid or second amino acid when you are going to analyze the fractions in the chromatography. So that's called C-terminal approach. All right. So are we clear up to this point? That how? Yes.
Why we need C-terminal analysis? As N-terminal is fine.
No, it depends upon the requirement of the people. Like if somebody is interested in finding CTDs like C-terminal domains, then instead of wasting their time and energy and reagent in sequencing the complete protein, they can go from C-terminal. So it depends upon objective, why you are doing sequencing, what you want to know. If you are interested in looking only at the specific part of C-terminal, then why would you waste your time and energy in doing that whole sequencing of protein?
Yes, sir.
Okay. So let me just take you again to the first slide. What we have done so far, we have seen the step number 4, this step, amino acid analysis. We have seen step number 2, that is denaturation and reduction. We have also seen the step number 3, that is terminal analysis. Now, the most important part, that is, one is proteolytic cleavage, and the next one is locating the disulfide bonds. So what did I tell you about proteolytic cleavage? When do we perform this proteolytic cleavage? The proteolytic cleavage has to be performed when you have longer peptides. Because if you have longer peptides like 100 amino acids, 200 amino acids, so it is not possible to go by this terminal sequencing methods. You cannot continuously do the sequencing from one end because they lose the sensitivity after a certain number of amino acid. So the best idea is that you fragment it in pieces and then you sequence individual things by terminal analysis. Okay, so you fragment that peptides and then you sequence this part, this part, and this part. Now what is the challenge here? In this picture it might look easy to you, that what is the challenge? There is no challenge. You split and then you sequence and you get a complete sequence. But as you will realize, in realistic situations, everything is present in a tube, right? So the moment you digest this peptide into pieces, all the pieces are present in a tube. And when you are sequencing it, you will get the sequence of this peptide, you will also get a sequence of this peptide, and you will also get a sequence of this peptide. Now, how will you assemble it? That's the biggest problem. And that challenge also comes in nucleic acid sequencing, because when you sequence complete chromosome, you break down that into small parts and then you analyze it. So how do you overcome that problem in DNA sequencing? In DNA sequencing, if you want to sequence a very long stretch of DNA, so you have multiple DNA molecules. Some of the DNA molecules are cleaved like this, then some may be cleaved like this, some may be cleaved like this. So you can take out these fragments, like you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11 fragments of different type. So obviously, what you can notice here, the sequence that you will determine in fragment number 2 will have some common part in fragment number 5, and it will also have some common part in fragment number 9. So this is how you overcome the problem of connecting the fragments or connecting the sequence, and then you assemble them. So if you have determined the sequence of number 1, then you can put it somewhere. Then you will see, okay, 4 is having some overlap with 1. So you will place sequence of 1, then you will place the sequence of 4, then you will place the sequence of 2, then you will place the sequence of 5. So this is how gradually you can connect the dots. And this in DNA sequencing, this whole process is very, very popular as chromosome walking. You walk across chromosomes by looking at what is the overlap area and what is the common area. If you are doing the PCR, you can do that by primer walking method. So that's connecting the dots, okay, joining the pieces by knowing that which part is common. Just like you solve some puzzles where you see this is the part of this, and then you might have also done some jigsaw puzzles where you just check for the matching parts, and then you connect those dots by looking at the matching parts and make the complete picture. So here you don't have that ability. I mean, how that problem will be solved? You should have fragments which should also have some overlapping parts. So if you can generate fragments which will have overlapping parts and you can individually sequence them, then it will be easy for you to assemble the whole sequence, isn't it? So we need multiple type of enzymes. What is the tool needed here? We need proteases of different types, which can do this function. Let's say in one tube, we digest with protease 1, and in another tube, we do digestion with protease 2. Okay, two type of enzymes. So what you will do by making this two type of digestion, the same peptide, let us say this is a peptide of some length, and this is same peptide in this tube. So since the proteases may be unique in their recognition site, so they will cleave at different positions. Protease 1 may have different specificities, and this can cleave this peptide in this manner, whereas protease 2 may have different specificities, and this can cleave in this manner. So don't you think in this case you will generate this type of sequences, and in the second case you will generate different kind of pieces? Now you can again go back for either of the type of sequencing and terminal sequencing. Now since you have sequences from two experiments, and you know that some part of this fragment, let us say 1, is matching with 4, some part of this sequence, that is 2, is matching with 5 here, and some part of the sequence 3 is matching with 6 here. So by looking at what matches, you can construct, you can say here the 1, this is 4, 4 is matching with 1. Then there is a sequence 2, 2 will have overlap with 5. Then you have sequence 3 that will have overlap with 6. So if you know both these sequences 1, 2, 3, 4, 5, 6, then you can say that the sequence present in 4 comes after 1, sequence present in 2 comes after 4. So just like with the help of this kind of overlap, you can connect the dots and you can get to know the complete sequence after proteolytic digestion. So just tell me yes in the chat if you understood what I mean to say. I'm trying to convey you that how to determine, how to match the fragments, how to know that which fragments comes after what when you have done the proteolysis and done the sequencing. Very nice. So let me now just take to other aspect. I mean, I will now show you a brief outline of different type of proteases, because when you are going to use proteases, you should know that what are their specificities. So since proteases are very, very important tools in protein sequencing, so you must have idea of proteases when you are talking about protein sequencing. Now basically proteases are also called as peptidases. They can be grouped into two categories. One may be known as endopeptidases, other may be known as exopeptidases. So just like you have endonuclease and exonuclease, exo means that cuts from the ends, and endo means that cuts within. Even in DNA, you might be familiar with some sequence specific nucleases which are called as restriction endonucleases. So you can say in endopeptidase category also, there are some peptidases which have preference for some amino acids. And why do they have preference for amino acids? You already know that an enzyme has got specific shape. You know that enzyme has got some shape of its active site. And the moment when they will interact with this peptide linkage, so the peptide linkage will also have a shape by virtue of amino acids. Every amino acid has got a different shape. So you can imagine that every proteolytic enzyme cannot have equal propensity to cleave every peptide bond, because the space between two peptide bonds will not be identical at every position. So this is why several endopeptidases have specificities. Okay, they have specificities. And on the basis of their specificities, what residues and what in what manner they cleave things, they have been also classified. So if I just describe you the overview or simple classification of proteases, then I would say that there are three major classes. One are called as serine proteases, another is known as cysteine proteases, another is known as threonine proteases. So remember that their name doesn't arrive from which amino acid they cleave, but the name arises from which amino acid is present in their active site, because that will decide what type of peptide bond they are going to cleave. So residue at active site that leads to this classification. So as the name suggests, serine proteases will have serine at their active site, and example of these kind of proteases are very, very popular digestive enzymes: trypsin, chymotrypsin, thrombin, elastase, subtilisin. These are all serine proteases because they have serine at the active site. Then comes another type of enzymes. They are called as cysteine proteases. So as the name suggests, as I told you, those enzymes will have at their active site which amino acid? Cysteine amino acid. So they are called cysteine proteases. A very common example, caspase. In fact, the name caspase arises from the fact C-asp. That means they have cysteine at their active site and they have aspartate at their active site. So those enzymes which have cysteine and aspartate at their active site, they were named as caspase. Similarly, papain is an example of cysteine protease. Tobacco H virus protease, scalpain is a very common blood protein, hepatitis virus protease. These are all examples of cysteine proteases. Then we have another major class that is known as threonine proteases. As the name suggests, they will have threonine at the active site. You should see one thing common, that most of the active site residues are polar in nature. Serine has got OH, cysteine has got SH, threonine has got OH. So the advantage is that if there are polar residues at the active site, then definitely they will be very, very reactive and they will be helping in this kind of proteolytic cleavage. So examples. So examples of these examples of, please keep your mics off, it causes unnecessary disturbance. Okay, so I'm muting everyone and you don't have permission to unmute yourself now, so there will be no disturbance. So what I was telling you, that besides these three major classes, serine protease, second one is cysteine protease, and third one is threonine protease, there are three more classes, that is aspartate protease, which use aspartic acid as active site. So a popular example is pepsin. Remember, don't confuse between papain and pepsin. Papain is a cysteine protease and it's found in papaya. Pepsin is your digestive juice. Then renin is also a digestive juice. HIV protease, cathepsin, priscillin. These are all aspartate protease. That's fourth category. Then these are some two rare categories, fifth and sixth, where metalloproteases, which use zinc or cobalt as ion present at active site. So a number of exopeptidases like carboxypeptidases are metalloproteases, and there are atom proteins in the matrix metalloproteases, also known as MMPs, which are involved in redox biology also. And then glutamate protease, which are equisulin, and there are some asparaloglutamic peptidases present in different organisms. So I would say these two, 5 and 6, are relatively rare classes, but those 4 classes which have been highlighted, they are all very, very abundant classes of proteases. So as I mentioned, there are plenty of proteases present. Okay, a number of different type of proteases present. Now, how do these proteases work? Do they have a sequence specificity? And if yes, how do they work? let's look at their specificities. So if you look in a simple manner, like I can give you one simple table to comprehend that. These are very, very commonly used enzymes in protein sequencing. And you can see here, there are few endopeptidases and few exopeptidases. So in endopeptidase category, you have a trypsin that has ability to cleave after C terminal of arginine or lysine. So what does that mean? That means you start a protein sequence writing from N side and you write from up to C terminal. So if there is any residue that is arginine or lysine, which means basic residue, basic amino acid, R or K, so this trypsin will be cleaving towards C terminal, or we also call it after. Okay. So after and before means whether it is a C terminal side or N terminal side. Remember, this side of arginine and lysine is known as N terminal side, this side. And this side is known as C terminal side. So if you say it cleaves before, that means it cleaves towards N terminal side of these two residues. If you say it cleaves after, that means it cleaves towards C terminal side. So as you can see here, trypsin has a specificity. It will cleave only whenever there will be R and K. It will cleave after that. So whenever you will have trypsin lysed fragments, they will always have at their ends either R or K. So if I just randomly make these peptides which are having R or K at their ends, that means they are trypsin digested products. Okay. Now similar to that, chymotrypsin has another specificity. What is their specificity? They cleave after aromatic amino acids. So if there are aromatic amino acids, that is like W, Y, or F, it will cleave after them. Pepsin, it is known to cleave after phenylalanine or leucine. Then elastase, this has this specificity. Endoproteinase, lysine, that has specificity of lysine. Endopeptidase V8, that has specificity of glutamate. Thermolysin has ability to cleave after any of these. So although there are plenty of things, I will tell you how you can explore that. You can have plenty of examples there. I will tell you the resource where you can find all these things. Similar to that, you have exopeptidases also. Besides endopeptidase, you also have exopeptidases like carboxypeptidase A and carboxypeptidase B. So remember their difference. Carboxypeptidase A and B both are terminal peptidase, C terminal peptidase. So remember, they will cleave always this last residue, this last amino acid. Now, what is the difference between A and B? When A will work and when B will work? A will work only when the last terminal amino acid is aromatic. Okay? So if it's aromatic, then carboxypeptidase A would cleave it, otherwise not. Okay, if something else is there except WYF, if anything else is there, then carboxypeptidase A will not be able to cleave it. And what about carboxypeptidase B? So it cleaves only if there are basic residues. I mean, if there are arginine or lysine, only then carboxypeptidase B would be cleaving here, otherwise not. So that's the difference between carboxypeptidase A and carboxypeptidase B. How do they cause a difference and how do they cleave? This knowledge should be there. I mean, you should be you should be having this prior information in your mind when you are looking at the problem solving of these kind of questions and the problems based on it. Now, besides enzymes, there are several reagents also, non-enzymatic reagents, non-proteolytic enzymes or non-enzymatic reagents, chemicals, which are also capable of causing a cleavage in peptide. Few examples are here you can see, like cyanogen bromide. So as you can see here, cyanogen bromide is capable of cleaving just after methionine. Formic acid can cleave after aspartate. Then these are two important components, bromosuccinamide and chlorosuccinamide. These can also cleave after tryptophan. And BNPS ketol, this is capable of cleaving after tryptophan. So some of the most popular things, I mean, I'm not going into long list. There is a very long list which you can find in the PDF that I will share after the class, which is associated with this lecture. So we should now focus on two most important things here. The first one here is cyanogen bromide, which is commonly used in sequencing. You should know that it cleaves after methionine. And in the previous category, the most commonly used enzyme is trypsin, that is known to cleave after arginine or lysine. Okay, sometimes people also use chymotrypsin, which cleaves after aromatic amino acids. So these are the most important thing which you should always remember. And then you should know the difference between carboxypeptidase A and B. B for basic, remember that. Carboxypeptidase B cleaves only when the end will have basic residues. Okay, A will cleave when it is not basic. I mean, it is not arginine lysine, then A will cleave. So these are highlighted things you should remember, and they will be very, very useful in your discussions or questions solving abilities. Now, one more last thing that I would like to tell here about the cleavage rules, that these cleavage rules fail sometimes. It is not necessary that if there is arginine or lysine and trypsin must cut it. It is also likely that trypsin may not sometime cut the bond even when there is a arginine or lysine. The reason is proteolytic cleavage is not only decided by the factor of what is present in near vicinity. As you can see here, the P1 and P2 are representing the cleavage site. So this is the amino acid. This is the position before the cleavage site. This is after the cleavage site. So let us say we are talking about maybe trypsin digestion. So this P1 may be arginine or lysine. And you know, if this is arginine or lysine, then you said a rule that it will cleave after this position. Okay. But what is the detailed rule? Detailed rule says that the cleavage is not only dependent on this P1. We cannot in a short form say that if there is arginine lysine, then after arginine lysine it will be cleaved. But it is also decided by what are the next 4 amino acids at the cleavage site and what are the previous 3 amino acids at that cleavage site. So they also have an influence on the abilities of cleavage. Reason? Can anyone tell me the reason? The reason is that enzymes may be pretty big. They may not be very small, like covering 1 amino acid. They may be quite big and they may have some active site, right? So they just take hold of the site, and an active residue, which we learned previously, that may be aspartate, cysteine, or glutamate, depending upon the class of protease. So active residue or active site amino acid will then interact with this peptide bond and will make a cleavage. But now, as you can see here, this bond, this protein is quite large, and this also covers a large area. So how well it is fitting into this space and how strong it is binding, it will be all decided by these adjoining amino acids. If these amino acids do not find this enzyme very friendly and they are not happy with this enzyme at any moment, so probably they will push this enzyme away. And if they push this enzyme away, then the propensity to cleave this particular bond will be reduced. So that's the reason why even after there is a residue where an enzyme can cut, there will not be cleavage. Okay. So what does it leads to, there is a very, very interesting thing called Keel's rule. What Keel's rule says, although there will be impact of other amino acids also, as I said, P1 to P4 on either side, but one of the most drastic effects happen if there is proline. Let us say this P2 here is proline. So what is the unique feature about proline? Proline is a very different amino acid. It has secondary amino group because its side chain, it should have been like this: carboxy group, NH2 group, H, and the side chain should be having 3 carbon atoms. But in reality, the amino group that interacts with the last carbon and that makes a linkage. So in reality, the proline that actually becomes like this. And this causes a lot of hindrance. So if there will be hindrance, there will be no flexibility in peptide that would not like sitting of an enzyme over that place. So proline will definitely cause a very difficult situation for any kind of enzyme that is coming and about to cleave this protein. And that is what is known as Keel's rule. Okay, Naveen, you have a question. Carboxy peptidase cleaves at C terminal after every amino acid except P, K, and R. No, I didn't get your question. Are you asking me, or please mention whether you are suggesting something or you are asking anything. I'm already telling you about the proline. So you can listen carefully. I'm mentioning about proline right now. So in case of the Keel's rule, what is the Keel's rule? This suggests that if there is a proline somewhere, I mean, this is the P1, this is the cleavage site, and if there is a proline sitting behind this, so this will just push the enzyme away. It will say, oh, I'm not happy with you, just go away. I don't like you because you cause a lot of discomfort to me. So that results in absence of cleavage when proline precedes in such cases. Okay, so what Keel's rule suggests, it defines the exception or non-ability to cleavage by trypsin if target amino acid, arginine or lysine, is succeeded by a proline. The commonly accepted rule is trypsin cut site as RK. So not only preceding, I was telling you about the earlier position, but even if this succeeds here, that will also cause a hindrance. Now, I will clarify that when succeeding proline will cause a defect, and when preceding proline will cause a harm. So since the active site here is this represented by green line, so now the most damage or most difficulty in binding of enzyme would be caused somewhere here. This is your active site residue, and just near active site residue there is proline. So hindrance would be more impactful at this place rather than P2. But remember, if some enzyme is N-terminal cutting enzyme, this is C-terminal cutting enzyme, that means it cuts after arginine or lysine. So proline that is succeeding will cause more damage. Proline after the site R or K will be more harmful, and that will not allow the cleavage. But if there is any enzyme that has the cleavage site here, let us say at N-terminal site, in that situation, presence of proline at P2 or preceding proline will cause more damage. All right, is that okay? So the presence of proline, in fact, you can see many other rules. This has been described here. This is your P1, this is your P2, P3, and P4. So you can say this is the cleavage site. So before the cleavage site, if there are different kind of residues like D, K, CD, C, K, H. So all these residues are representing they are favorable. And those which are not favorable, that is primarily proline. So up to 4 residues on either side can decide the propensity of peptidal cleavage. And that means every time the cleavage will not be equally efficient. Cleavage may not be equally efficient at all the times. So are we clear about these rules? Let me just take some simple problems for you to understand so that you get to know what I was trying to describe to you. Let me just take a couple of simple problems first. So let's see this particular question. A very simple question. You have a tryptic digest of heptapeptide built from 3 lysines. Okay, there is a peptide which is made up of 3 lysine, 2 alanine, 1 tyrosine, and 1 phenylalanine, yielded tri and tetrapeptide. So remember, how would somebody know that there in a protein there are 3 lysine, there are 2 alanine, there is 1 tyrosine and 1 phenylalanine? They will know by a method that we described, amino acid analysis. So all right, they are just asking in this question, if a peptide is made up of these things, then which of these 4 options is correct, provided that on digesting with trypsin, the peptide yielded a tetrapeptide, a tri-amino acid peptide, 3 amino acid peptide, and a tetrapeptide. So first look at this thing, that which is the peptide that is fulfilling this rule. 3 lysine, 2 alanine, 1 tyrosine, and 1 phenylalanine. I think everyone. So you cannot make a choice on this basis. But yes, if you now look at the trypsin rule, so where does trypsin cleaves? Trypsin cleaves after lysine and arginine. And remember, trypsin is endopeptidase, so it will not cleave on the first bond. Although here is lysine, so this bond will not be cleaved because this is terminal bond. This bond will also not be cleaved. This is terminal bond. So where do you think there are lysine in between? This is 1 lysine in between. Okay. Then you cannot expect in this case, because it is a terminal bond, then you can have 1 lysine here. So you have in these 2 cases, similarly you have 1 lysine here which can be cleaved by trypsin. So now look at the results. This is already omitted out because even if you digest by trypsin, just a moment, you cannot eliminate this. This can also be cleaved because you have a lysine here and you have a cleavage site at this place. So if you cleave these things by lysine, this will be cleaved here, this will be cleaved here, this will be cleaved here, and this will be cleaved here, right? So since you can see here, this will give you a dipeptide and pentapeptide. This will also give you a dipeptide and pentapeptide. This will also give you a pentapeptide and dipeptide, but this will give you a tripeptide and a tetrapeptide. So what has been asked in the question fulfills this criteria, and therefore this is the correct sequence for this given question. Let's look at one more thing. Which peptide bond marked as A, B, C, D, and E will be broken when the following oligopeptide is treated with trypsin? So again, you have the same example. That means you know the trypsin specificity. Trypsin cleaves at the C terminal of arginine or lysine. So you remember that trypsin after RK. So there is a sequence: lysine, arginine, proline, lysine, arginine, glycine. Now you can mark the probable sites. So you have a lysine here, you have an arginine here, you have a lysine here, and another arginine here. So these are the possible amino acids which are recognized by trypsin. But where the cleavage will take place, let's see this. Do you think at the first site cleavage will take place? Yes or no? Just tell me. Will the cleavage take place at the first position, A? No. Why not? Because it is the first bond or terminal bond. And trypsin is not a exopeptidase. Will cleavage take place here? No. This is also terminal bond. Cleavage will not also take place here, right? But if you talk about this, will cleavage take place here at B? It is not a terminal bond. Again, no. The reason is Kills rule. You have a proline succeeding arginine, so that will prevent the cleavage at this place. But yes, this site doesn't have much problem, doesn't seems to have a greater problem, although there will be some slowness in the reaction, or the degree of reaction, or propensity to break down, or speed or pace will be slow, but still this bond can be cleaved. So you can have a cleavage at this bond. Therefore, what has been asked in this question, which bond A, B, C, D, E will be broken when the oligopeptide is treated with trypsin at pH 7? So of course, this is the right answer. Okay. Let's now look at another problem. So consider the structureless oligopeptide. There is an oligopeptide which has a sequence given here, quite a long sequence. The number of bonds that will be cleaved by trypsin and chymotrypsin treatment separately are. So little difficult. You need to know here where trypsin cleaves and where chymotrypsin cleaves. So you should know the rule. Trypsin cleaves after R and K, provided that there is no proline succeeding, and chymotrypsin, it cleaves after aromatic amino acids, that is tryptophan, tyrosine, and phenylalanine. It is important to remember the single letter code of amino acids to be very efficient in that. So we have to know these things. Now let's see. First we talk about trypsin digestion. So let's highlight the trypsin cleavage sites. You have arginine. Let's recognize where arginine and lysine are present. So R, K, and one R here. And I don't see anywhere else. So what do you say about these sites? Just make an evaluation. Will this bond be cleaved? No, because this is terminal. Will the first bond be cleaved? No, this is also terminal. Will this bond be cleaved? Can this bond be cleaved? Yes or no? Yes. Okay. Because there is methionine succeeding this, and proline is much later than that, so it doesn't cause much difference. It doesn't completely inhibit the cleavage. So yes. So how many how many bonds will be cleaved by trypsin? One bond. And now if you go to chymotrypsin, let's see that. So let's check the chymotrypsin sites and look for aromatic residues here. So you have tyrosine here, you have tryptophan here, phenylalanine here, and I don't find any other aromatic residue. So let's check whether these things can be cleaved or not. Tyrosine will be cleaving here or not? Tryptophan will be cleaving here or not? Tell me about these two things, yes or no? Yes, both positions fine. Glycine is always happy because glycine is very flexible. It says, oh, please come on, cleave this. I'm happy to welcome you. But proline is very rigid. It says, no, I can't change myself. You should not come here. So proline doesn't allow the cleavage, but glycine can do that. So cleavage will happen here. Cleavage will also happen here, but not at this place. Why? Because this is a terminal residue, and chymotrypsin is not an exopeptidase. That will be done by exopeptidase. So there will be only two bonds which will be cleaved by chymotrypsin. So you have this as a correct choice here, 1 and 2. Now let's look at one more interesting thing. Similar kind of thing. You can answer it very easily. The oligopeptide is given here is treated with trypsin, chymotrypsin, and carboxypeptidase B. So little more complex. They're giving three things to you. So let's now use the same rule, first rule. The sites of two things are already given to you, trypsin and chymotrypsin. So let's check for trypsin recognition sites, R, and there is no K. So do you think this site will be cleaved? Yes or no? Can trypsin cleave at any of these two sites? No. Apply the Kiel's rule. Proline says, no, you cannot come here. I don't allow you cleavage. So no at these two sites. Now let's talk about chymotrypsin. So chymotrypsin, you have to find out where are aromatic residues, F, F, and there is no aromatic residue in between. So again, will this site be cleaved? Can these things be cleaved by chymotrypsin? No. These are terminals. This is also terminal. And here you had Kiel's rule. So not yet. Trypsin cannot cleave, chymotrypsin cannot cleave. Now let's say about carboxypeptidase B. So what was the rule for carboxypeptidase B? B for what? Basic residue. I mean, if you have a basic residue, that means either arginine or lysine, only in that case it will be able to cleave this, but there is no basic residue over here. So when there is no basic residue at the terminal, do you think carboxypeptidase will be able to cleave? No. So net answer is, irrespective of you add trypsin, you add chymotrypsin, you add carboxypeptidase, still this peptide cannot be cleaved. So there can be some small peptides for which you have to choose really different non-conventional enzymes. Trypsin, chymotrypsin are very popular enzymes for sequencing, but you cannot use them for certain peptides like this. So this was just to practice, how these rules work. Now coming back to our idea of solving the problems of protein sequencing. I mean, how do you get the complete sequence? How do you assemble the whole thing? If you remember my first slide, let me also take you once again to the first slide. So this was my first slide where I showed you different steps. And we talked about protein purification, step number 2A, 2, 4. But we are left with now two steps, that is locating disulfide bonds and then complete sequence determination or integration of the information. So with the help of two problems, we will try to look at this, that how to connect all the dots and how to then get a complete protein sequence. So some very interesting questions I have which can decipher or help you to find out the disulfide linkage positions. Interesting question. So what does this question say is, look carefully. It says that there is a 30 residue peptide which was treated with trypsin, and tryptic peptides were separated by HPLC. So you digested a peptide by trypsin and then you took that and separated that by HPLC. And what you saw in HPLC, there were 4 peaks, A, B, C, and D. Now peptides corresponding to A, B, C, and D were then reduced and alkylated alkylated selectively at cysteine. You know how to do that. You can add iodoacetamide and then you can prevent cysteine from reverting back. So that's a way of preventing disulfide bonds to be formed again. The sequence obtained from A, B, C, D after reduction and alkylation there were 4 peptides. So for all those 4 peptides, they determined sequence. And let's see what are the sequences. They say that sequence of one of the peptide is AEK. Sequence of the second peptide is C. I'm just putting star for representing alkylation. EPGYR. And then there was 4th peak, which was corresponding to WC star SPPK. And then there was 4th peptide, that was C. Sorry, it was C star. Or maybe I have one diagram. I will help you. Instead of writing that, I'll do that. I'm highlighting it here. So there were 4 different peaks A. When they sequenced it, the sequence was found to be AEK. Then they got B. What B had, B gave rise to one peptide CEPGYR and WC SPPK. So this came out from the second peptide. Then third signal that came from a peptide, what was the sequence? C star EFGR, C star GGK, and finally they had another peptide D which gave rise to C star EAFC and this. So what do you understand from it? Let me just know. This is a method to determine the position of disulfide bonds, right? So remember that what they have done, understand the procedure. First, what they are doing, they are just digesting the protein without cleaving the disulfide linkages. Okay, let me just draw it for a representation. So suppose you have a peptide and you have one disulfide bond here, one disulfide bond here, and one disulfide bond here. So what you are doing, you digest without breaking SS bond. And when you do so, let us say there is a cleavage site here, there is a cleavage site here, there is a cleavage site here, and there is a cleavage site here. So don't you think it will break down into pieces? It will make a fragment. This should be fragment number 1, A. This should be your fragment number 2, that is B. This should be your fragment number 3, C. And this should be your fragment number 4, D, and this may be E. So once you do digestion without breaking down disulfide bond, what will happen? Although the digestion will take place here, it will rupture this place, it will break down this place, but don't you think fragment A and B will still be connected together because you have not broken down the disulfide bond? So A and B will be together, and since you have a disulfide bond formed between this area and this area, so although digestion will take place here, here, and here, C and D will still be connected by disulfide linkage. And finally, you have a piece that is E, since it has a disulfide bond only within its own region, it doesn't make any disulfide bond with other fragment, so it will be separate with disulfide bonds broken down. So now what they did, the procedure says they are digesting it without breaking disulfide bond, and then they are adding some reducing agent and alkylating agent like iodoacetamide. So what does it suggest? It suggests to you that if you want to determine the exact position of disulfide bonds, you first digest the peptide without breaking disulfide bond, and then you separate them. So these fragments can be separated by chromatography. CD will be at different position because of size, AB will come together, and then E will come together, and then you determine, after breaking down their disulfide bond, you determine their individual sequences. So if you came to know that in your first fraction A and B were together, that means there was a disulfide bond between these two things. If you came to know that C and D came together, that means there was a bond between C and D. But what would have happened if there was a disulfide bond between this place and, let's say, the last place? So you would have had A and E together during your analysis. So this is what the logic of understanding and finding out the disulfide bridges. Now let's look at this problem. Okay, selectively look at this problem. So maybe you can check the sequence of all the peptide is same. The only thing is you have different locations of their disulfide bridges, and you have these fragments given to you. So it is quite obvious. What you can see from here, you can easily answer this question. AEK, since it was separate, it doesn't have any disulfide bond. So AEK will be separate. Fine, it is separate, same everywhere. Now, C star or alkylated C, EPGYR and WCSPPK, they came together in one fraction. That means these two pieces were connected by disulfide bond. That's why they came together. So we have to look in all these four options that where do you find a disulfide bond between these sequence. Let's highlight this sequence. So I am highlighting this EPGYR. This is your EPGYR. This is your EPGYR here. This is your EPGYR here in all the four options. And then where is WCSPPK? WCSPPK is here. So just look at the situation. They have shown the four different possibilities of different type of disulfide bonds, but you can see that in first case, this fragment and this fragment is not connected. But according to the results that we got, since the fraction B had these both things together, this part and this part should be connected by a disulfide bridge. So in which of these options you can see that this part and this part, blue part, shaded part, is connected together? Which option do you see? Just check and answer me in the chat. In which option the blue shaded part is connected by a disulfide bridge? That's only in option number 3, right? You can see here that in option number 3, there is a disulfide bond that is connecting this blue region with this blue region, but not in any other. If this would have been true, then there would have been 1, 2, 3, 4, 5 different fractions, right? Here, in this case, this region is connected with the last one. So this peptide should have been with EACFL. They should have come together. So this was a trick to answer that none of these things would be right, but this is correct because this is connected here. Similarly, you can further evaluate this. You have your green colored region, that is your EFGR. This is your EFGR. This is your GGK. And this is your, the next fragment is this, EAFCL. So what does this indicate? Since you have got EAFCL separately, it is not bounded to anything, but these two green shaded areas are connected together. So you should also have a disulfide bond between these two green areas. And as you can again see in the second option, in the third option, I'm sorry, there is a disulfide bond here between these two green areas. This one is connected with this, okay? But the last one, D fragment, was alone. It has got 2 cysteine in itself, and it is not connected with anything else. So this is how, by doing this simple procedure that I mentioned, you digest the protein and then you fractionate it by chromatography and then you reduce it, and after reduction you do the sequencing, then you can get the exact location of your disulfide bond by connecting the dots. You see which fragments have come together and then you put them together. You write a disulfide bond between them. Then you see which two have come together like this. You make a disulfide bond between them. And then by looking at tracing the sequence, you can make them in particular order. Okay, so this is how we determine the position of disulfide bonds in a peptide. And we came to know that this is the correct position of disulfide bond. So your point number 5 is clear. While sequencing peptide, how to decipher position of disulfide bonds. Now, one last thing, I would just exceed it slightly by 5 to 6 minutes. And I will tell you how to integrate all this information. Suppose you have done amino acid analysis. Suppose you have also done the fragment analysis and trypsin digestion. How will you integrate the information to determine the exact sequence? So last point, that let us see that with the help of a question. So the question says, in order to determine primary structure of an octapeptide, that means a sequence, what they did, They did amino acid composition analysis. That means what I told you as a step number 3, AAA, amino acid analysis. They hydrolyzed everything and then they found how many amino acids are present. So this was one of the experiment. Just keep a note of experiments. Experiment A is amino acid analysis. What is experiment B? Treatment with carboxypeptidase. So they treated and digested with carboxypeptidase. That was done in separate tube. Remember that these are 4 different experiments which have been done in separate tubes. So what is your third experiment? Third experiment is digestion with chymotrypsin. And your experiment number D is digestion with trypsin. And your experiment number E is digestion with CNBR. So I hope you remember the whole idea why we are doing this digestion with multiple enzymes. Because if you have one protein sequence, you can create the fragments in different ways. Suppose red color is indicating your chymotrypsin digestion, green color is indicating your trypsin digestion, and your yellow color is representing CNBR digestion. So what you will get after this, in one case, you will get tryptic digest. This is how you will get tryptic digest. Let's say I name them as T1, T2, and T3. In another tube, when you do chymotrypsin digestion, since there are 2 sites like this, so this will break down into 2 longer fragments and 1 smaller fragment. And let me name them as C1, C2, and C3. And when you do CNBR digestion, you break that into 2 parts, and let me write them as CN1, CN2. So idea of doing these 3 experiments together, and maybe carboxypeptidase also, is to fragment these peptide in 4 different ways. Why we are doing that in 4 different ways? So that we can walk upon one fragment over another. If we know the sequence of this fragment, this complete fragment, which was digested by, let us say, cyanogen bromide, and another fragment that was digested by chymotrypsin. So once we know this sequence, we can have some common region between this and tryptic digest. And therefore, by just doing a way like chromosome mapping, we can do here a walking of peptide, and then we can connect the sequence and we can get an overall sequence of these things. So that's why we do multiple digestion, and we look at the results of each experiment. So just see what they did. They did, amino acid composition analysis of each of these experiments, and they did different things. So look at the results. A, B, C, D, E. These are results of each experiment. When they did amino acid analysis, they got this. When they did carboxypeptidase digestion, they got this. And when they have done chymotrypsin digestion, they got this. When they did trypsin digestion, they got this. When they did CNBR digestion, they got this. All right. So although if you don't have a choice like which is given here, having a choice makes your life easier. But if you don't have choice at all, like let us say I don't have 4 options A, B, C, D, then what would I do to find out protein sequence? Then I will first look at the amino acid composition. So I know that there are 2 serine, there are 2 alanine, 1 arginine, 1 lysine, 1 methionine, and 1 phenylalanine. So I will make some random combinations on a sheet of paper, because at least I know that there are 2 alanine, 2 serine, 1 phenylalanine. So I will just make random combinations on a sheet of paper, and then I will write down. Okay, this is the first thing that I will do. All possible sequences I will write down in a notebook. And then I will check the results of all those things, carboxypeptidase, chymotrypsin, and then I will tally with all my options that in which case, in which sequence, I can get these kind of results. So here, instead of all possibilities, they have just given 4 possibilities. In fact, you can have several different possibilities. So let us say there are these possibilities which I have pointed out on sheet of paper, and now I want to determine complete sequence of a peptide. So let's check one by one. What do you know about, let's begin with trypsin, because you are very familiar with trypsin by now. So what trypsin does? Trypsin cleaves after arginine or lysine. Remember the Keel's rule, remember the terminals. So just mark out arginine and lysine and see what is the result of arginine lysine. When you digest it by trypsin, you get 3 different pieces. In one piece, there was alanine arginine. In another piece, you had lysine, phenylalanine, and serine. And in third piece, you had alanine, methionine, and serine. So let's check in all these 4 choices that which of these 4 options would give me this particular result of trypsin digestion. So let's see where trypsin will digest. Let's try to find out that trypsin will digest after lysine or arginine. So I just marked lysines. I also marked these arginines. These are also possibly cleavable. Since this arginine is present at border, I will not consider all these things. So I have marked the possible arginines. Now check which one matches with your experimental outcome. If you cleave your peptide here by trypsin, do you think 3 peptides will be obtained? Yes or no? No. You cannot get 3 peptides. You will just get 2 peptides. So that means your assumption that your sequence is this goes wrong. This is incorrect. But if you cleave your peptide here, do you think you will get 3 peptides? Again, no. So this is also not right. I mean, if this would have been right sequence or this would have been right sequence, then you should have got only 2 peptides here, but it is showing 3 pieces of 3 peptides. So these 2 possibilities are gone, right? Now we are left with these 2 possibilities. You can check here if it is digested here or it is digested here. We can get a fragment which is having methionine, serine, and alanine. Yes, we have that. One fragment that is having serine, phenylalanine, and lysine. Yes, we have that. Another fragment, alanine, arginine. Yes, we have that. Right? Now, if this would have been true, here also, there is a difference or interchange in these amino acids. So this is also a likely answer. So still, with the help of trypsin digestion, we cannot come to a conclusion that which of these 2 options, 3 or 4, is correct answer. Now let's look at another thing. Let's talk about chymotrypsin digestion. So where do you think chymotrypsin digests? Chymotrypsin cleaves at the C-terminal of aromatic residues. So let's look at aromatic residues. These 2 choices are already ruled out, so we will not consider them anymore. We will now be talking about the remaining 2 choices. So where are your aromatic amino acids? Phenylalanine. You have phenylalanine. Okay. These are aromatic amino acids. So let's check what would happen. If you cleave this peptide, option number 3, with phenylalanine, what you are going to get? 2 peptides. What you see in actual results, when you digest with chymotrypsin, yes, you get 2 peptides. And what are the amino acids in these 2 peptides? The first peptide has got alanine, arginine, phenylalanine, and serine. Just look here. Alanine, arginine, phenylalanine, and serine. Yes. In the second peptide, lysine, methionine, serine, and alanine. Yes. Lysine, alanine, methionine, and serine. Fine. But this is not going to help you. If you look at the second choice, I mean, this is resembling your results. But if your cleavage would have happened here, if the 4th option would be true, then chymotrypsin digestion will give you a peptide that will have alanine, arginine, and phenylalanine, only 3 residues, and another fragment that will have 5 residues. But you don't have those results. You have 4 residues in each of the fragment. So that means, since you did not get the desired result by chymotrypsin, this option is also incorrect. So you are left with only one option that is right, and that seems to be third. So since you had options here, choice here, you were able to filter out it easily. But if you don't have choice, then what you do is you actually make use of your random probabilities and you draw the sequence. There are some computational tools which can generate random peptide sequences for you. And then from there, you can filter out and you can get and assemble the complete sequence. So that completes the idea of protein sequencing. Okay. Now, let me just have a recap quickly. We have a couple of steps, like if I go back to the same slide, let me just revise that thing for you. So first step is protein purification. You purify a protein, then you denature it, reduce it, and after denaturation reduction, you also have to make sure that your mercaptolithanol has been added, and then disulfide bonds have been acetylated or methylated so that they don't form again by using iodoacetamide. Then you do proteolytic cleavage if the peptide is large by trypsin, chymotrypsin, cyanogen bromide, and then you perform N-terminal analysis to get the sequence. And then at the same time, if you want to locate disulfide bridges, then the strategy is you first cleave or digest with peptidases, and then you separate them by HPLC, and then you do the sequencing. And simultaneously, you can also do acid hydrolysis, and you can know the amino acid composition, and then do chromatography to finally assess the different things. And we then looked at how we can combine this whole information to get a complete sequence of a peptide. So this was something that fetched Frederick Sanger a Nobel Prize. That's not a small thing, seems to be easy for us, but provided the conditions or the available information at that time and the availability of technology, that was a tremendous work. And that was a foundation which was laid by him so that we are able to sequence not only peptides, but also proteins, but also nucleic acids. There are more than 100,000 proteins which have been sequenced so far, and several hundred thousands of genomes have also been sequenced. So with that power, we are able to now decipher the relationship between a lot of things, that is relation between the sequence to phenotype to genetics. So molecular biology, biochemistry, genetics, everything is well connected with each other because of these two breakthrough technologies. One is protein sequencing and other one is nucleic acid sequencing. So that's all from my side. I will just open the session for a couple of minutes of discussion and then we will stop.